Hi again. So today I'm going to be talking a little bit more in detail about some of the characteristics of the macromolecules introduced in the last lesson. Okay. And so we're going to look specifically at the monomers of each of the macromolecules. Okay. And so a monomer is a single component of the macromolecule as a whole. Right? They're kind of like the building blocks. So if you think of a big building, a big school such as this one, right? An individual brick, as a brick, is a part of that overall structure. These are the bricks that build up the macromolecules. Yeah. And so the carbohydrates, their monomers are called monosaccharides, and they're singular rings. If you put together two single rings, two monosaccharides, you get yourself a disaccharide. You put together more than that, you get yourself a polysaccharide. And so you just link a bunch of rings together and you get different carbohydrate structures. Okay? Monosaccharides. As we look at lipids, you'll see here in my beautiful model, a fatty acid. So the monomer of lipids are fatty acids. For the most part, actually that's true. It's always a fatty acid, and usually they're this chain shape that you see here in the model. There are some lipids that take on ring forms, and we'll look at those when we talk about the functions of lipids, but they're still composed of a base of a fatty acid that's just changed configurations. Okay? And so the fatty acid has a carboxylic acid group in the front, right? A carboxyl group, and then a long chain of carbon and hydrogen. Okay? And so these peaks, whenever you see these peaks drawn like this in any kind of organic chemistry, each of the pointy peaks is a carbon. Okay? So this is 1 carbon, 2 carbons, 3 carbons, 4 carbons, 5 carbons, 6 carbons, 7 carbons, 8 carbons, 9 carbons, 10 carbons. Okay? And now if I was drawing this accurately, it would keep going and going and going. They're very long. Okay? Nucleic acids are the next one, and the monomer of nucleic acids are nucleotides. All nucleotides have 3 main components: the phosphate group, which is, not surprisingly, comprised of a phosphorus with some oxygens attached to it. Okay. This phosphate group is linked to a sugar ring, and so it's going to be ribose or deoxyribose if we're doing RNA or DNA. And then there's this other group on the end here, and this is the nitrogenous base group. And these are variable. Right? So depending on what nucleotide you're looking at, you're going to have a different nitrogenous base. Okay? We have adenine, cytosine, guanine, thymine, and uracil. You probably recollect those from your past experiences in biology and dealing with DNA and RNA. Okay? And last and not least is our proteins. The monomer of proteins are amino acids. They have 3 groups. We have the amine group, which is the front of the amino acid, basically. It's a nitrogen with some hydrogen stuck to it. You have the really fun and cool functional group. The functional group has this variation here that we represent with an R, right? There's no element with an R on it. You can't see my periodic table, it's over there. But there's no element with an R on it. Right? It's a functional group, it's variable. In living things, we have 20 different amino acids that can contribute to the structure of proteins. So this R group can be 20 different variations, and depending on what variation we have a different shape and therefore a different function for this amino acid. Right? And then on the butt end of it, we have a carboxyl group. When we take these monomers, right, so if I take monosaccharides, I always link a monosaccharide to a monosaccharide. I'm not linking a monosaccharide to an amino acid. Right? I'm linking them with their appropriate monomer friends. We link monomers together and we get a polymer. Mono means one, poly means many. Right? So we create these long chains of different monomers to create different macromolecules. The chemistry that achieves that is a dehydration synthesis. It's called dehydration because it's removing a water from the 2 monomers. Okay? So you get monomer 1, you get monomer 2, you put them together, right? Usually with some enzymes or some energetic situations. Okay? An OH from one of them will pop off, an H from another one will pop off, join up, make an H2O, that flies out. Okay? And then the spaces where those got ripped off from link together. Okay? It's a dehydration synthesis. And so here I've used some amino acids to demonstrate this. So I have amino acid number 1 right here. I have amino acid number 2. Okay? And so from the carboxyl group, okay, my OH is popping off and flying down. Okay? And then from my amine group, an H is popping off. Okay? And then after that, you'll see that my 2 groups are now linked. Right? So this and this got linked. Okay? The H2O flies away, bye water. And we have ourselves a nice, cute little dipeptide at that point in time. Okay? And then that would happen on this end. It would just keep building and building and building and building and building. You get thousands of these together, you get yourself a protein. Okay? The opposite of this is hydrolysis, right? And so if you add the water back in, you can rip apart those monomers. Now, building up the larger molecules requires an energy input, right? That's a decrease in entropy, which is a concept that we'll have to talk about and I'll have to describe. I think I'm going to probably make a whole video on entropy. But anyways, so if we decrease the entropy by making something more organized and bigger, right, that requires an energy input. It can't happen spontaneously. However, if we are increasing the natural entropy of a system, right, that releases energy, that can happen spontaneously. Right? And so that's why, like, if you leave a carbohydrate out in the world, it's eventually going to break down into little pieces. Right? And so hydrolysis can occur spontaneously in the presence of water. Yeah, it's the opposite of dehydration synthesis. Break stuff down. Okay? And here is a cute little chart. This cute little chart shows you the monomers and the polymers of each, right? So I take some monosaccharides, I shove them together with dehydration synthesis. I got myself this big, sexy carbohydrate, right? 3 monosaccharides stuck together. It's going to be more than 3 usually, but sometimes it's 2. Disaccharides form a function, right? Glucose is good to go. I think that's a monosaccharide, so yeah. Then we got fatty acids, right? You link them together. And now if you look at this one, you can see when you link together the fatty acids, they link together out here on this third party, right? And so you can get these glycerides, right? So this is a glycerol component, and you link them all together, and this would be a triglyceride because you have 3 fatty acid tails. You can build bigger ones too. But then you got nucleotides. You link them together at the phosphate group to one of the carbon groups of the next ring. Dehydration synthesis occurred. And then amino acids I showed you already. You link them together, you get proteins. Cool? So yes, monomers, link them together through dehydration synthesis, you get your polymers, which are the functional macromolecules. Cool? Bye.